My name is Riya Grandi. I'm a ninth grader um, and a rising sophomore, and this is my thesis defense. In the overview, I'll be going over the title, introduction, objective, methodology, and conclusion. The potential permanent treatment of Harlequin Ichthyosis through gene editing, nanotechnology, and non-viral vectors. Introduction. Harlequin Ichthyosis is a dermatology-related disease caused by a mutation in the ABCA12 gene. Excessive skin is produced in the newborn as a result of the mutation. Various conventional therapies are used to treat it. However, there is no permanent solution to this treatment currently. Harlequin ichthyosis, as previously mentioned, is a skin-related disease caused by an inherited recessive gene. Some conventional therapies include bathing regularly, a lot of nutrition, medications, and creams to clear scaly plates formed on the skin from this condition. Objective. The sole purpose of this proposed research is to develop an efficient method to deliver a functional ABCA12 gene into the target cell and restore the function of the skin using various methods. Some of the methods discussed in my paper are the difference between gene therapy and gene editing. Gene therapy can assist in the treatment of Harlequin ichthyosis as an efficient enhancement of the skin, but it's not permanent, it's temporary. That's why it's irrelevant to the potential permanent treatment. But if it was used in this case, augmentation would be the most practical. However, even though the gene is dominantly expressed over the defective gene would still be inherited in the process of keratinization, which is the reproduction of skin cells. But it can still assist in gene editing, particularly in non-viral vectors, because non-viral vectors have a lot of negative effects in the skin, but we can enhance the skin using gene therapy, which is why I still talked about it in my research paper, even though it's irrelevant for the permanent treatment. And non-viral vectors are basically vectors that deliver substances, not using viruses. So these examples in this case could be sanoparesis, which is the use of sound waves to penetrate the cellular membrane, and reversible electroporation, which is using electrical pulses to penetrate the membrane. And another main one is microneedles, which are small substances at the nanoscale that penetrate the skin barrier in order to deliver the necessary components. Oh yeah, and however, the skin being the largest organ in the body is difficult to permanently restore. Due to this unfortunate complication, small areas of the skin will have to be treated, but hopefully in the future with developing technologies it can be resolved. Methodology. This is a proposal and not a statistical analysis, I have collected information for the comparison of various techniques used elsewhere. These comparisons include using sanoparesis, electroporation, and microneedles at the same time, the combination of them. And I needed to research that because I didn't want to risk the combination of them and have negative effects on one another. And I have not provided specific results due to the rarity of this condition and inaccuracy using other skin conditions. These include applications to penetrate the cellular membrane, inorganic, organic nanoparticles, and not viral and non-viral vectors in order to deliver this CRISPR gene editing tool. And the reason why I chose a proposal instead of a statistical analysis is that when I was first choosing this research topic, I came across Harlequin ichthyosis, and I wanted to do it because it's a rare condition and it's really hard, and there's not a lot of research on it, and it also contributes to my dermatological interest and a potential career I want in the future. But there wasn't a lot of statistical data that I could rely on, so I decided to do a proposal. In conclusion, I've concluded that the use of non-viral vectors such as sanoparesis, reversible electroporation, and microneedles is necessary to penetrate the cellular membrane. The disruption of the structure will assist the transportation of the necessary genetic substances through inorganic and organic nanoparticles. The delivery of functional CRISPR-Cas9 gene editing tool is necessary for the treatment. CRISPR-Cas9 is a gene editing tool used from a bacterial defense mechanism. It precisely modifies an organism's DNA by adding, removing, or altering specific genetic sequences at targeted locations. The delivery of this tool to the defective keratinocytes is essential to the enhancement of the skin, and I'm aiming to deliver CRISPR-Cas9 in keratinocytes, specifically in the stratum basal, because that's where the production of keratinocytes occurs. And nanotechnology is another major factor in this proposal, because nanoscience consists of two types, organic and inorganic. Organic nanoparticles are used to encapsulate the genetic material to protect it from external effects, and inorganic nanoparticles are designed to identify and target cells. However, the skin, being the largest organ in the body, is difficult to permanently restore. Due to this complication, small areas will be treated. Thank you so much, Gifted Gabber. I would like to acknowledge the Gifted Gabber professors for their assistance in my research. The motivation and effective advice really helped my journey. Thank you so much for the amazing experience. I'd like to thank Professor Lilova for the articles and specific on advice on exactly what to research, and Virgil for the encouragement and advice on the methodology.
Awesome job. You can stop sharing, Riya. Really good topic. Let's have Professor Lilova ask you some questions. Okay, you can stop sharing.
Great work. I just want to know that this is extremely complicated topic. It's really difficult one. And there is not one answer to how we are going to treat it. It's a very complex mechanism. So I still would like to ask about your opinion. When we are trying to deliver the genes, right, the CRISPR gene editing tool, what carriers do you think would be most suitable for that?
Based on my research on nanotechnology, I think organic nanoparticles, because they can more effectively sustain the genetic material. So yeah.
And biocompatible, of course, which is always what we are trying to achieve. Great. It's a very, very difficult topic there. Yeah, great job.
Yeah. A question for Riya, Professor Virgil.
Yes, Coach. Job well done, Riya. I have one question. What do you think are the strengths and limitations of your research?
So there's a lot of limitations because, as mentioned before, the skin is a large organ and is hard to work with. But I think the strengths include now we have like specific things on what to research, and it could develop more technology and research on the specific advancement.
Thank you.
Great job. Yeah, fantastic. So thank you for being here.